All right, folks. So here we are, and we're doing our final video here. Just wanted to show you the outline. Again, we had our intro. We did hydrocarbons. We did carbohydrates, lipids, proteins, and finally now we're on to nucleic acids. This was my cheat sheet of the outline, and I did post that on our information, our classwork tab. So make sure you print this out. It's a nice little cheat sheet to remember everything by. All right, so let's get into nucleic acids. Now I have to admit, I did a video and it totally messed up. So this is my second try. So you're going to see writing on these things, and hopefully that writing won't interfere with what I'm explaining. Okay, so now we're down to the nucleic acids, the last of our monomers and polymers of our biomolecules. Okay, this time nucleic acids, the elements involved here are your carbon, hydrogen, oxygen, nitrogen, and the new one on the block is the phosphorus. Nitrogen was in the proteins, carbon, hydrogen, oxygen was both in lipids and in carbohydrates. So now we have phosphorus. There are two types of nucleic acids, DNA, RNA. We're going to see a comparison of those two. We know DNA provides the directions, which makes RNA, which then eventually goes out to a ribosome and makes a protein. Good old central dogma of biology. DNA makes RNA, which makes protein. DNA can be found in the nucleus of eukaryotic organisms, or it's floating around in the cytoplasm of a prokaryotic organism, such as bacteria. RNA, though, can be found in a number of places. And I know when you think of RNA, you probably think of mRNA, tRNA, rRNA, but there's lots of other types of RNA out there. And you probably even heard RNA in the news with the coronavirus vaccine, how they're making an RNA vaccine. All right, so let's get started a little bit more with this monomer polymer thing. The monomers of nucleic acids, there's my beads, right, are called nucleotides. So what is a nucleotide? What is the structure? Just like amino acids had a certain structure and they were the monomers of proteins, what's the structure of a nucleotide for nucleic acids? And when we think of these nucleic acids, we're either going to have DNA, which is a double strand that's twisted of these nucleotides, or RNA, which is just simply a single strand of these nucleotides. Okay. You're going to see a lot of differences here, like in talking about the nucleotide, you're going to see the differences between DNA, but everybody remembers Mrs. Horner's beat of remembering what is a nucleotide made up of? Phosphate, sugar, and a base. A phosphate, a sugar, and a base. Now, there are these adjectives on some of these, like a nitrogenous base. What does that mean? It means that it has nitrogen in it. And a pentose sugar. What does that mean? It has 5 carbons to it. But the phosphate group, you should know, that's one of our functional groups out there. And that's what's in one of these beads. Each bead for a nucleic acid has a phosphate, a sugar, and a base. Okay? Now, what are the bases? The bases come in 2 forms, what we call pyrimidines and purines. Pyrimidines, I always say, are a smaller molecule. They're like 1 ring big. But purines are like 2 rings big. They're a little bit bigger. That's what this is all saying. A single 6-member ring. This has a 6-member ring fused to a 5-member ring. In other words, 1 ring and 2 rings. Okay? 1 ringy dingy, 2 ringy dingies. Then the pentose sugar. If it's DNA, it's called deoxyribose. If it's RNA, it's called ribose. And we'll see what the difference of those are. And then finally, the phosphate group. So phosphate, sugar, and a base. Okay, let's look at the diagrams of this. Now, this first picture is going to show you a simple diagram of DNA versus RNA. So let's first look at the nucleotides of each. Here's a DNA nucleotide. It's made up of a phosphate, sugar, and a base, right? Here's the RNA nucleotide, phosphate, sugar, and a base. So far, it looks the same. Phosphate here, still the same. But here's where it starts to get different. This sugar here in DNA is called deoxyribose. This sugar here in RNA is called ribose. Why? What is the difference between them? And it's right there. See that right there? There's an OH. Here, it's only an H. It's deoxy over here. Aha, that's where the name deoxyribose actually came from. It's a ribose sugar, but it's deoxygenated. It doesn't have that extra oxygen there. That's hence the name deoxyribose, okay? So it's just that difference between DNA and RNA. Not much, but it's there. The next difference between them is this base. Down here, we see the bases in DNA can be A, G, C, or T. But the bases in RNA can be A, G, C, or U. Why U? When U attaches as a base here, it can attach. But if T tried to attach here because of that oxygenation there, it can't do it. Okay, that's the bottom line picture. Now, what's this? The other difference between DNA and RNA is DNA is double-stranded, RNA is single-stranded. See here, I circled each nucleotide here. Phosphate, sugar, and a base. Phosphate, sugar, and a base. Phosphate, sugar, and a base. Yep-a-da-ba-da-da. Okay, phosphate, sugar, and a base, but it's all single strand. It's just a single strand of those things. Notice it could have an A, could have a C, could have a G, could have a U, could have a whole bunch of U's if it wants to. Have a whole bunch of G's. Okay. DNA, on the other hand, it's double-stranded, okay? So we get into this whole base pairing stuff. Because, yeah, we have the phosphate, sugar, and a base, phosphate, sugar, and a base, phosphate, sugar, and a base, Oh, wait a second, though. This phosphate seems to be upside down, okay? That gets into this 5', 3'. You might remember that from first-year bio. If you don't, we'll remember it. Now, A always matches with T. We got to think why. And I'll get back to this diagram, but I'm going to show you this diagram here, okay? This is a more intricate diagram of DNA. Sorry, my printer kind of smudged it here. Don't worry about that over there anyway. What we want to do is we want to look at this nucleotide right here, phosphate, sugar, and a base. Look at this base. It has 2 rings. Is that, I'm going to hide this up. Is that a purine or a pyrimidine? And it's a purine because purines are 2-ring big. Here is a single ring base. That's a pyrimidine. Okay, so phosphate, sugar, and a base. Here's the phosphate, sugar, and a base over here. Okay, notice that this one seems to be like upside down. Okay, why A with T? Okay, and we'll get to that upside down thing in a moment. Why A matching with T? Because a purine has to match with a pyrimidine, right? That's one reason. A double ring has to go with a single ring. See, double ring with a single ring with a double ring with a single ring. Always has to be a purine with a pyrimidine. But if A and G are both purines, and this is A, why does A have to go with T? Because C is also a pyrimidine. Couldn't we put C there? Couldn't we put the cytosine up there? Why does thymine get to pair up with adenine? And here's why. In between adenine and thymine, there's 2 hydrogen bonding sites, 2 with a 2 here, okay? But in between cytosine and guanine, we have 3 hydrogen bonds, okay? You have to have a 3-prong with a 3-prong, a 2-prong with a 2-prong. It's kind of like those plugs, you know how they have 3 prongs or 2 prongs, okay? You don't want to stick a 3-prong in a 2-prong outlet. It just won't fit. And sticking a 2-prong in a 3-prong outfit, well, you know, it just got an empty space there. Could be dangerous. Okay, so we know that this is labeled adenine, this is labeled thymine. And if you look at the colors, you can figure out what's what. But what if it's in black and white? Let's flip this over. Okay, let's figure this out. Okay, here's, don't worry, it's like multiphosphated up there, but here's your phosphate, sugar, and a base. Notice it's a 2-ring and it's a 1-ring, and there's 2 bonds. Well, A always has 2 bonds between T. C always has 3 bonds between G. So since this is 2 bonds, then this has got to be A and T here. Which one's the A? Which one's the T? A is the purine, so this is the A. This is the pyrimidine, so this must be the T. How many bonding sites here? 3. Is it A and T or is it C and G? It's got to be C and G. Now here's the double bond ring here. The double ring, is that the cytosine or the guanine? It's the guanine, okay, because guanine is a 2-ring purine. So this is the cytosine, which is 1 ring. Here's 2 rings. So is this A and T or C and G? 2 hydrogen bonds, 2 hydrogen bonds. So is it A and T or C and G? Right, it's A and T. Here's 3 rings or 3 hydrogen bonding sites. So this must be C and G. Which is which? Here's the purine. Here's the pyrimidine. Since this is the A and T. A and T. Here's the purine. Here's the pyrimidine. This must be the G. This must be the C. Okay? Now, get back to one other thing. This upside down thing. Why is one side of the DNA molecule pointing in that direction and the other side is pointing in the opposite direction? We call this the 5' end and this the 3' end. So this is the 5' end and this is the 3' end. What is this 5', 3'? Well, there's the Mrs. Horner simple way of remembering it. I always say, look at the sugars. They look like little houses with a roof, right? And I always say, find where the O, the tip of the roof, that's 5, that's at the tippy top. Okay, so this is the 5' end, and then down here would be the 3' end. But what does this 5 and 3 actually stand for? At each corner of this house, right? We're just looking at another house here. At each corner, plus this little bend up here. These are all the carbons in that sugar. Remember, we said it's a 5-carbon sugar. 1, 2, 3, 4, 5. Well, you number them always like going clockwise around. 1, 2, 3, 4, 5. 1', 2', 3' is down, 5' is up. That's where they actually get the 5' up here, 3' down here. And again, 1, 2, 3, 4, 5. 5 is up, 3 is down. Over here, though, start at the O, go clockwise. 1, 2, 3, 4, 5. 5 is pointing down, or you can use a Mrs. Horner like, hey, let's look at the house here. The house, the roof is pointing down, so that roof is always the 5. Okay, so that's the structure of DNA and RNA. We saw the comparison of the two here. And the final thing I want to show you, okay, with DNA and RNA is the central dogma. Kind of like a picture of that going here. Okay, here's DNA, and this would be in a eukaryotic cell because DNA is inside the nucleus. DNA through transcription makes RNA. And some of you might remember that that's called transcription. Okay, we'll talk about that later. You don't need to know that term now. Then that RNA leaves the nucleus, it comes out here, and through the next process of translation, that RNA will be translated into a protein. That's the central dogma. DNA makes RNA, makes protein. DNA makes RNA, makes protein. DNA makes RNA, which makes protein. How does DNA make RNA? That is transcription. And how does RNA make protein? That is translation. That is the central dogma. And a dogma is like a mantra. It's the most important thing in biology. All right, so let's sum up everything we've done now in this unit, okay? And I call this the old Cho-Cho-Chon-Chomp chart. And why do you call it the Cho-Cho-Chon-Chomp chart? Because you start off with the first column, which is what are the elements in carbohydrates? Cho, C-H-O. Okay, that you can't see too well, so let me try and get a darker marker here. Okay, what do you see in lipids? You also see Cho, okay? Proteins are Chon. And I'll let you fill in nucleic acids, okay? So Okay, function, carbohydrates. You got to tell me later. You got to fill in what the function of carbohydrates are. Lipids, I'm going to say long-term energy. This is the energy that your bodies are going to turn to after all the carbs are out. So, sounds like the carbs must be some immediate energy. Okay. Lipids, long-term energy. Proteins, tons of functions. Look them up, fill them in. Nucleic acids, basically one function for nucleic acids. DNA makes RNA makes protein, which is the genetic information. Genetic info. You can fill that in as well. Cell parts involved. Cell parts. What makes carbohydrates? What breaks them down? Look that up in your notes. Lipids. We said phospholipids are a big important part of a certain cell part. So when we think of lipids, this cell part that's in ER and in a vacuole and in a Golgi apparatus and even in the cell plasma boundary around this cell thing, that's the cell part that's involved in here. But proteins, I always like